Good morning to you all. Today we start with a new chapter, which is Streptococci. By the end of this chapter, you as students will be able to tell the following, or to achieve the following objectives. Explain the characteristics. What are the main characteristics of the Streptococci in general? Explain Lancefield classification, or we call it Lancefield grouping, and this is a grouping that is based on the carbohydrates and proteins on the surface of the bacteria. So peptidoglycans, they classified this bacteria based on the presence and absence of these epitopes, let's say, immunologically speaking. Identify clinical infections. What are the infections and the diseases that are caused by Streptococci? Compare streptolysin O with streptolysin S. These are toxins that hemolyze blood, one of them in the presence of oxygen and the other in the absence of oxygen. We'll talk about this in details inside this chapter. Explain variance factors for different species. What are the factors that cause this bacteria to be harmful? What are the arms and the weapons that this bacteria uses to make its living and to harm us as hosts? These are the bacteria. So genera and species to be considered. We are going to talk about the strep that is of a clinical relevance or medical importance to us humans, and not all of them. We will talk about mainly 3-4 maximum. If you see here, we have beta hemolysis, alpha hemolysis, and gamma, which is no hemolysis. So of the beta hemolytic Streptococci, we have two important species: Streptococcus pyogenes, which we call group A, the Lancefield grouping, and Streptococcus agalactiae, which we call group B, which is also Lancefield grouping. These are the most important and they cause diseases to human beings. In the new edition of the book, which is 15th edition, they added Streptococcus dysagalactiae, group A, C, G, L. We are not very much interested, but good to know. Alpha hemolysis, it's also important. One of them is important, which is Streptococcus pneumoniae. And then we have pseudopneumoniae. Also, this is something new in edition 15. We have also alpha hemolytic bacteria, which we call viridans Streptococci. Viridans means green. Viridans Streptococci or viridans Streptococci, alpha hemolytic. And we have 3 types of them. There are more, but these are the ones we are interested in: Streptococcus mutans, salivarius, and metis. Then we have Streptococcus bovis, another group. And finally, we have the enterococci. These enterococci, in the 15th edition of this book, Bailey and Scott's, they have grouped them into 1 to 5 groups. Group 1, 2, 3, 4, 5. Based not as in the Lancefield grouping, which is based on the carbohydrates and proteins on the surface of the bacteria. No. This is based on what? 16S ribosomal RNA gene sequencing. So it's based on phylogenetic analysis. 2 of these enterococci are important to us: Enterococcus faecalis and faecium. And usually these 2 are tested for food contamination. If you have a food or any contamination coming from the sewage, then we test for these. If we have these 2, faecium and faecalis, in high quantities, high amounts, then the water or whatever you are testing is contaminated with sewage. Understood? So far, so good. So these are the ones. Pay attention to the 3 red ones, maybe a little bit to the enterococci. But the 3 red ones here are our focus and our interest and and should be of importance. Pyogenes, agalactiae, pneumoniae. Many of these Streptococci normal microbiota. They reside in the pharynx, inside your mouth, deep inside pharynx, mouth, outer part, lower GI tract, intestines, lower, vagina, especially agalactiae, the group B, and may be pathogenic if we have the following conditions. Depleted microbiota. Normally, as human beings, we have normal bacteria, which we now call normal microbiota or microbiome. We have them inside our gut, and they are very important for our life. They produce vitamins like vitamin K. They also overwhelm and kill the pathogenic bacteria, so we need them for our life to be okay. So they are vital. Sometimes, due to many activities the human beings do, let's say using lots of antibiotics, or drinking, let's say, what we call them artificial sweeteners. Coca-Cola, you have aspartame and acesulfame. These are diet cola and zero cola and these stuff. They have artificial sweeteners. And others, maybe chocolates, maybe other food products that have artificial sweeteners. It has been found that these artificial sweeteners, they affect the normal microbiota. And of course others. So if you have depleted microbiota, these Streptococci, which are supposed to be normal flora, they will activate themselves and become pathogenic. Impaired bacterial density. It can be your microbiota is down or the Streptococci is more than usual. Increased variance. Bacteria, they are variant, pathogenic. But in certain circumstances, you will see that this species or this strain of the species, it's more pathogenic than the rest, is more aggressive and more virulent for many reasons. A mutation in the bacteria is usually the main reason. Let's say you have Streptococcus pyogenes. If it's very virulent, it can cause necrotizing fasciitis, which is eating the flesh of the human. Impaired immunity. Normally, as human beings, we have strong immunity. If you get older, then your immune system will become weaker. Then the bacteria, the opportunistic bacteria, the normal flora, which we have in our bodies, they will become more opportunistic and they will cause problems to you. Or sometimes you have a disease like HIV, or you may have kidney transplant where they have the doctors lower your immunity to minimize the rejection of the new kidney. In these circumstances, these bacteria, they become active and they become more and more opportunistic and cause problems to us. Gain access to sterile site. These are the fluids and the sites in our body that the count of the bacteria equals 0. Normally should be 0. First, blood. Our blood is bacteria free or organism free. CSF, pleural fluid, peritoneal fluid, pericardial fluid, bone, joints, bone and joint fluid, vitreous fluid, and vascular fluid. All of these, if the bacteria gains access, it will cause problem. These are the areas which Streptococci are most commonly encountered in. Vagina, Streptococcus agalactiae. Usually the ladies who are pregnant, before delivery, they are tested for the presence of agalactiae. Why? Because they might be transferred to the newborn and cause a problem to the newborn. GI tract, gastrointestinal, upper and lower. You see upper, look here, upper in the pharynx or the nasopharynx cavity, or in your intestine, upper and lower. Skin, S. pyogenes, skin infections. We will talk about or the skin diseases caused by Strep pyogenes. Respiratory tract, RT, You have in the upper part, you have S. pneumoniae, which is in 50% of the cases, or most of the cases, it's normal flora. S. pyogenes in the upper respiratory tract. S. pyogenes, it's never microbiota, never. So if it's found in your nasopharynx, it will cause pharyngitis. And there are other diseases, sequelae diseases, that follow pharyngitis if not treated. We will talk about them in this chapter also. So we have Strep pneumoniae up here, and then we have Strep pneumoniae down here in our lungs. So if we have Strep pneumoniae in our lungs, most of the time or all the time, it's pathogenic, and it causes a disease called pneumonia. That's why Streptococcus pneumoniae, and there is also Klebsiella pneumoniae. This is the habitat where you might find this bacteria and the mode of transmission. S. pyogenes group A, so it's not normal flora at all. Don't expect to find this as normal flora like others. It's in skin, upper respiratory tract, nasal, pharyngeal, and mucosal. Mode of transmission, person to person. Hugging, shaking hands, sometimes coughing and sneezing, airborne, indirect. Then agalactiae, group B. You can find it as normal flora. Female genital tract, vagina, lower GI, upper RT, respiratory tract here, occasional. Is this agalactiae the same, normal flora? S. pneumoniae, that's also another one which you are very much interested in. Normal microbiota maybe in 50% of the cases in the upper respiratory tract, but we could test you all here for S. pneumoniae, and you could all be easily negative. But if it's there and you are not feeling, or you are feeling okay, then it's normal flora. It's there and it's not making any harm to you. No damage. Coughing and sneezing, secretions, pathogenic if it's in the lower tract, in the lungs, I mean. Viridans group, the green one, the same thing. Normal microbiota, oral cavity, GI tract, GI tract, gastrointestinal, and female GT tract. Endogenous strain, it can be there originally. Into sterile sites from dental manipulations. This can cause problems. If you go to the dentist, it's in your mouth and there's some sort of a wound during the dental manipulation. By mistake, can introduce this into your blood, then a problem happens. As I promised you, we are going to talk about these three mainly. So these three here are the most important, or the most medically important bacteria in this chapter. S. pyogenes, or group A, and here it says Lansfeld grouping based on carbohydrates. See, carbohydrates on the cell wall. And this causes upper respiratory tract infections, such as pharyngitis, very common cause, and then rheumatic fever. Acute pharyngitis, this is the one. Skin impetigo, lesions and ulcers on your skin. And I will show you a picture as we go along in this chapter, exactly similar to the impetigo caused by the staph in the last chapter. Erysipelas, I will show you a case of erysipelas. Always significant whenever isolated, a normal flora. Every time S. pyogenes is found, then it's pathogenic. That's a strep, you know. Strain or a chain of balls, Streptococci or cocci. Agalactiae, group B, main cause of septicemia, meningitis, and pneumonia in neonates during delivery. This tells you why we kept all the time testing the ladies when they are pregnant. We don't want this to happen to the baby. Septicemia, bacteria in the blood of the baby, that's septicemia. Meningitis, inflammation of the meninges in the brain, and pneumonia, lung disease. And S. pneumoniae. If you look here, we never said it's Lansfeld grouping. Can you see? No Lansfeld grouping. So it looks that S. pneumoniae is ungroupable. They didn't make grouping of this. So you don't say S. pneumoniae group B or group C. It's pneumoniae, it's separate. Not like A and B. A and B, group A, group B, and there's group C, G, D. So this one is not groupable. 95% of the bacterial or pneumonia. The pneumonia is the lung disease. Main cause of bacterial meningitis. In the human being, we have meningitis sometimes as a disease. And it can be caused by any bacteria that can get access or gain access to the CSF. However, it was found that the most commonly encountered bacteria that cause meningitis in a human being are 3. That doesn't mean others are not, but these are the 95% of the cases. What are these 3? Strep pneumoniae, Neisseria meningitis, and Haemophilus influenzae. These are the 3. 3 of these bacteria, they comprise 95% of the meningitis cases. 5% is the other bacteria. So S. pneumoniae is very important. Therefore, we are vaccinated against these 3 bacteria. Causes other diseases caused by pneumonia: otitis media, inflammation of the ear, not the external ear but the intermediate ear; sinusitis, inflammation of the cavities of the nose. These are all other diseases caused by pneumonia or strep pneumoniae. Let's continue with the pathogenesis. Strep pyogenes, streptolysin O, that's the first variance factor. Oxygen stable. So if there is oxygen, it can work. Non-immunogenic. It doesn't produce or it doesn't elicit immune response in the human being. Does not elicit antibodies. Cytolysin. It destroys the cell, the human cell. This is a toxin that destroys our cells, the host cells, such as RBCs, white BCs, neutrophils and others, and platelets, thrombocytes. This cytolysin, usually called hemolysin, destroys these RBCs. SLS, streptolysin S. This is oxygen stable. There is the other one, which is streptolysin O. This is oxygen labile. When it's exposed to the oxygen, very quickly it's destroyed. However, this is immunogenic. The body will produce antibodies against this particular toxin, and we call these antibodies ASO, anti-streptolysin O. There was a test in serology which is called ASOT, anti-streptolysin O titer. So this toxin destroys red blood cells on the 5% sheep blood agar plate, but under one condition: no oxygen. Therefore, this here can be used in the lab for diagnosis. This one, streptolysin O, it's immunogenic. It produces antibodies, which means if you can detect these antibodies by serology, then you can say we have this infection. That's why we use this in the lab in a test called ASOT. There is the SPE, streptococcal pyrogenic exotoxin. It's erythrogenic toxin, and this toxin produces red rash. We call this disease scarlet fever. It's one of the diseases produced by this bacteria. Heat labile, super antigen, the inflammation or the cytokines or the interleukins that are produced during this inflammation or this infection, it is really exaggerated in large amounts. Why? Because it's super antigen. It's an antigen that is big and causes high inflammation response in the human being. M protein is a helically coiled, helical polypeptide, which means this is a protein, DNA helix, intertwined polypeptides, protein, and that they are anchored where? In the cell membrane and the cell wall, so they do not detach. So this M protein is antiphagocytic. Anti-M protein cross-reacts with hard tissue, causing endocarditis. C5a peptidase. Peptidase means what? It's an enzyme. It inactivates chemotactic factors. If you look here, so this is the infection caused by strep, group A, and then the body calls for help which is neutrophils to come from all over your body and come to the site of infection to fight the bacteria there. What does this C5a peptidase do? It interferes in this call and prevents these white BCs from coming to the site of infection. We call this inactivating the chemotactic factors, the calls for help, thus inhibiting phagocyte recruitments. So you will not see any white blood cells coming to the site of infection for help to destroy the bacteria because of this C5a peptidase. So these are the enzymes produced by the S pyogenes. Deaminase, like arginine deaminase, maintains bacterial biofilm. So deaminase maintains bacterial biofilm. It helps the bacteria to build a biofilm on the tissue so it can stick there and make the harm for us, of course. Streptokinase, fibrinolysin, digests fibrin that kills S pyogenes, but streptokinase prevent this. Hyaluronidase, it's another enzyme that destroys the connective tissue, and then your tissue gets loose. DNase destroys the DNA. If you see C5a peptidase, which we just explained, destroys complement factors and chemotactic factors. Protease degrades chemokines, hindering phagocyte recruitment, the call for help. So these are the enzymes, the most studied enzymes that S pyogenes use as variance factors. That's just a quick thing to have a look at. Group A carbohydrates, these are the surface proteins that are variant factors. Promote survival, that's one. Protein F induce adherence, can stick easily to the surface, making biofilm. M protein inhibits phagocytosis and complement. And also this M protein that produces antibodies can also have non-specificity, or we call it cross-reactivity with heart tissue and cause problems there. Hyaluronic acid capsule, HA, evade immunity and promotes survival. SOF, serum opacity factor, invades cells. It's just for you to know. You don't have to know all of these. FBI, fibronectin binding protein, inducing adherence and anti-phagocytic. All of these are proteins that have been studied and found to cause all the functions we just mentioned. So it's not strange to find 2-3 proteins here on the surface doing the same thing. Promote adherence, anti-phagocytic. And then we have LTA, lipoteichoic acid, form biofilm. Lipoteichoic acid also it's commonly studied also, like M protein, these two. Forming biofilm. If you ask me, is this all? No, there are others, but these are the most studied. Then we have the toxins, streptolysin S, streptolysin O, lyse cells, any cells, red blood cells, white blood cells. Streptococcal pyrogenic exotoxin induces inflammation. So this bacteria has a good set of weapons against us. Enzymes, protease, degrading chemokines. Hyaluronidase, digesting tissue. Streptokinase, lysing fibrin. As I said, we made use it as humans. We made use of this as treatment for MI patients sometimes. DNase, destroying DNA. Deaminase, maintaining the biofilm of the bacteria. So all of these are variance factors, whether it's a carbohydrate on the surface or protein on the surface, whether it's a toxin, produced toxin, and or whether it's an enzyme. So enzymes, toxins, and protein or carbohydrate on the surface. These three things that are usually various factors for this S. pyogenes. Let's go to S. pneumoniae. S. pneumoniae, it has pneumolysine. Pneumolysine is just like hemolysin, but they called it pneumolysine because it's produced by the Streptococcus pneumoniae. Pneumolysine, pore-forming toxin, activates complement, suppresses oxidative burst. Our immune system, some cells, they have oxidative burst, H2O2. They can be secreted and they go to the bacteria and they kill the bacteria. This pneumolysine suppresses this function, evades immune system and lyses RBC. So it has many functions, this pneumolysine. Capsule, many bacteria are capsulated, and the capsule is a layer outside the cell wall. And usually this layer is sticky, slimy, and mucoid for two things. The main is to make biofilm, adherence, and then make biofilm and antiphagocytosis. See, you have cell membrane, then you have cell wall, and then you have the capsule outside. So 3 layers surrounding this ball or cocci. Phosphorylcholine, adherence to tissue and entry into the cell, facilitates entry of the bacteria inside the cell through binding to platelets, activating factor receptors. So they use the receptors that are normal to platelets, and then they use these receptors to get access inside the cell, and then make the damage, destroy the cell. Phosphorylcholine, pneumolysine, and polysaccharide capsule. That's just a quick thing. Cell wall, thick, and then the capsule polysaccharide. Shields from phagocytosis. This is the pore, like a tube, and all the toxin produced inside goes outside and released. So this is pore-forming toxins. Pneumolysine activates complement, suppresses oxidative burst, evades immune system, and lyses RBCs. Autolysin, lytic amidase. Cell lysis, again, autolysin. Pili or pili. This makes the bacteria maybe move faster. Yes? Promotes adherence and inhibits phagocytosis. Again, it's like the capsule. PCHO, again, this phosphorylcholine, adherence and entry inside the cell by using the platelet receptors. This red one here, pneumococcal surface protein A, if you remember we had protein A in the staph, protects against complement. Pneumococcal surface adhesion A, aids in cell invasion. Pneumococcal surface protein C, protects against complement again. So these are coming out. Bacteriocin inhibits growth. That's another toxin. Inhibits growth of the tissue or our cells. That's another one. Neuraminidase, cell adherence and survival. A third one, IgA protease, destroys IgA, which is an immunoglobulin. Lipoteichoic acid enhances inflammation. So as you see here, each bacteria has an arsenal of weapons. We call these, in medical term, variance factors. Okay, let's go to the diseases. So look at the diseases here. Streptococcus pyogenes, group A, or we call it gas, group A strep. Acute pharyngitis, that's number 1. This bacteria gets access inside our mouth, and then it causes tonsils here become, if you go down here, it's white. Sometimes it's even clearer than this. So all the pus is seen inside. If you are affected here with acute pharyngitis and not treated, there are sequelae and other diseases that come as part of this disease. We'll talk about them in this chapter. Look, impetigo, streptococcal impetigo. Look how it looks. It looks like lesions with crust, see here, and these are streptococcal. Of course, you have to make from this here, from the clinical picture, it helps you, but you cannot tell, is it streptococcal or staphylococcal? So you have to make tests, maybe culture, maybe other tests, or PCR, and then test and check if it's Streptococcus pyogenes or Staph aureus, this or that. This is cellulitis. This is the normal hand, and this is the change in color. So this is cellulitis. And here you have erysipelas, little bit of swelling and redness. Erythematous swelling, we call it. Look at this. This is necrotizing fasciitis. This is flesh-eating disease. After, let's say, in a hospital, they make an operation for you, and in the hospital there is this pyogenes and this strain in particular, which is very vicious, very aggressive strain, and if it infects your wound, this is what happens. It eats the flesh and your wound becomes black, necrotic. This is caused by Streptococcus pyogenes and sometimes Staph aureus even. Myositis, inflammation of the muscle. See here, you have this fluid-filled papule, myositis, inflammation of the muscle. This is caused by streptococcal toxic shock syndrome. And as you see here, of course, to give accurate diagnosis, you need tests at the end. Scarlet fever, the rash. See, red rash on the trunk, the chest, and even on the tongue. Strawberry tongue, yes, thank you. So scarlet fever, strawberry tongue. Scarlet fever. Pharyngitis. Look, that's what I told you when I said that if you have acute pharyngitis and severe like this, look at the pussy patches here, pussy areas, and even the tonsils are very big, yeah, inflamed and swollen, largely swollen, with patches of pus here. So if you just keep it without treatment, then rheumatic fever will happen. Antibodies will be produced against the M protein. What happens then? Immune complex will form, cross-react. So this is an immune complex, a group of antibodies together. That's immune complex. And where does this immune complex go? This immune complex cross-reacts with our heart tissue, or our skin, or our brain even, or our joints. So if you have pharyngitis, then rheumatic fever, then you wait a little bit, antibodies form against the M protein, which we talked about, immune complex form, and this immune complex cross-reacts with tissue of some organs in our body, including heart, skin, brain, and joint. This is dangerous. That's why pyogenes is dangerous. Pharyngitis, but not only pharyngitis, other diseases. It goes here, endocarditis. It goes to the skin, erythema marginatum. It goes to the brain, chorea. It goes to the joints, arthritis. See how many diseases come after the pharyngitis? Many diseases. We call them post-streptococcal sequelae or post-infection sequelae, diseases that come after. Okay, we stop for today and we will continue next week. Any questions, ladies? No questions? Okay, let's meet ourselves in the lab in 15 minutes.